You're going to be happy to hear that this is going to be a very short lecture. We've been talking about enzymes. Remember, enzymes can speed up your chemical reactions. Now, sometimes you need to shut down a reaction. Those enzymes are making products, and eventually you can get an abundance of that product, and it would be wasteful to continue to make more. So you need to be able to shut down this process or shut down the enzyme. We're going to look at two ways we can inhibit or turn off enzymes. We have competitive inhibition and non-competitive inhibition. Let's take a look at the competitive here. We know that substrates can go into the active site of an enzyme. So here's the enzyme. And then the substrate can be converted into the product. Well, we have a problem because we have a different molecule, an inhibitor, that is occupying already the active site. Therefore, the substrate can't fit into the active site, and then the reaction in the enzyme is being inhibited. So it's competitive because they're going after the same thing. Both molecules want to attach to the active site. Let's take a look at non-competitive inhibition here. We have two molecules. They're not going for the active site, though. The substrate would bind to the active site, but there's a different site over here on the enzyme, and this is called the allosteric site. That would be the allosteric site. When this inhibitor, the non-competitive inhibitor, binds to the allosteric site, you can see what happens to the active site here. It changes its shape, and the substrate can no longer bind. So we've stopped the reaction using a non-competitive inhibitor. If you think about it, this molecule is not competing for the same spot. It's not going for the active site, but it can bind to a different site, altering the shape of the active site and inhibiting the reaction. Now I'm going to go back to the top of this, and we're going to relate that to what we call a biochemical pathway. When you're making a molecule, usually it doesn't occur with just one enzyme and one step. Sometimes it does, but sometimes it takes many enzymes to convert A to B, another one to convert B to C, another one to take C and turn it to D, and then finally we get our product. Let's say that this process is continuing to happen and the body is making a lot of histidine. So let's go ahead and put a lot of histidine in there. If you want to go ahead and label them HIS, so you know we're making a bunch of the product. Again, these molecules, they don't have brains. There has to be some kind of signal to shut these things down, but they can't think, hey, we need to shut down this process. So the process of shutting down these reactions is called negative feedback. When you get an abundance of some molecules, they can feed back, which means go back to the beginning here, and they can hook on to the enzyme and shut it off. It can happen in a couple of different ways. So let's say that we have, I'm going to draw this enzyme right here. So let's draw enzyme 1 in this process. And this is the substrate, you know, this is A. So let's make an enzyme that kind of has that shape, like this, okay? And enzyme 1 is supposed to take the substrate and then convert it into B, which we have right here. That's what that diagram is showing you. Well, what can happen is the product, when it becomes more abundant, there's more of it, then it can interfere in this reaction. Histidine, when there's a bunch of it, can start to occupy the active sites. And again, we already talked about when that's happening, that's competitive inhibition. So let's write competitive inhibition. Negative feedback is occurring when you have an end product that can feed back and inhibit an enzyme early in this process. So this is a process that we see here. And so this is an example of how competitive inhibition can lead to negative feedback. And so if this enzyme is being inhibited right here, then you're not going to get any B, and then you're not going to get any C, and so on. You're not going to get any more histidine. As a matter of fact, you won't get histidine until all the histidine is used up, even this one right here. And now it's not going to be able to inhibit anymore because it's all gone. And so the reaction or these processes will start back up. Now, it might not just be competitive inhibition. Let me show you just the other option here. I'm going to draw, again, I'm going to draw the enzyme. And histidine might bind to this allosteric site. So we can put histidine here, so then it changes the shape. So sometimes this negative feedback is done by non-competitive inhibition. It just completely depends on which pathway in the body we're talking about.